Yes, there are a lot of slides, but not a lot of information because the slides are a lot of slides are just to explain the process of muscle contraction as well as later on cell movement. Okay, so don't be scared by the fact that there are 40-some slides that we have to go through today. So let me remind you what we're doing. We're looking at another example of convergent crosstalk, basically, right? How do the different muscle cell types carry out or are told to carry out muscle contraction? It has to do with hardwiring. So all the thing about hardwiring and signaling and convergent versus divergent crosstalk, that's what we're doing right now. Okay. So in terms of skeletal muscle, how are skeletal muscles hardwired, and then how do they perform muscle contraction? We have to look at these three things right here. For skeletal muscles, the contraction is voluntary. And whenever we say voluntary versus spontaneous, or another word for spontaneous contraction is involuntary, right? So it has to do with your central nervous system. Voluntary means that you think about it, right? And if it's spontaneous or involuntary, that means that doesn't involve the central nervous system. That's basically, at the end of the day, that's what that means. In either case, signaling still has to occur. So spontaneous or involuntary doesn't mean that these cells aren't signaled to undergo contraction. So for skeletal muscle, it is voluntary. It's all or nothing. And what does it mean by all or nothing? All or nothing means when you hear all or nothing contraction, you think about the fact that there is a huge amount of calcium ions being leaked into the cytosol, right? And all of the myosin heads are interacting with the microfilaments all at once. So that's all or nothing. Versus graded means that there are different levels of calcium ions that are being leaked into the cytosol, and then the number of myosin heads that are told to bind to microfilaments are variable. So it's not. So that's graded. So again, all or nothing means very high calcium ions in the cytosol, and all of the myosin heads have to bind, versus not. Right? Performance for skeletal muscle cells, it's a very high power, but it's not a constant performance. Okay, so again, it's voluntary, but it at the cost of fatigability. So now let's look at how skeletal muscle cells are hardwired to do that, and this is how it is hardwired to do that. Okay, pay attention to this right here. Is the fact that the dihydropyridine receptor is found in the T-tubule. Okay, the thing though is that the for skeletal muscle cells, the dihydropyridine or DHP receptor in the T-tubule is not directly linked to a calcium ion channel. That's very important. And the other receptor that is important is that ryanodine receptor. The important part is that the ryanodine receptor is a calcium ion channel. So notice this. Notice the fact that the ryanodine receptor is in the membrane of the ER, or the sarcoplasmic reticulum, right? So we saw this picture. So what happens when skeletal muscle cells have to contract? You have a signal from a neuron activating the plasma membrane of the skeletal muscle to undergo action potential, right? So a signal. The action potential will then move through the plasma membrane down the T-tubule where the hydropyridine receptors are. But then the hydrogen DHP, short DHP, it might be activated, but it's not connected to calcium ion channels, right? Therefore, that signal then is relayed into the sarcoplasmic reticulum. So the action potential goes from the plasma membrane to the T-tubule to the membrane of the sarcoplasmic reticulum. This is where the ryanodine receptors are, and ryanodine receptors are calcium ion channels. So therefore, when the action potential reaches the sarcoplasmic reticulum membrane, then the calcium ion channels are open. And therefore, for skeletal muscle cells, the calcium ions that are being leaked into the cytosol comes from the sarcoplasmic reticulum, not from the extracellular matrix. Right? That's important. And number one, because they leak in huge amounts, that's why it's all or nothing, right? So all the calcium would bind to the sarcomeres, and that's all or nothing. But the other is that fatigability. What happens if the calcium continue to leak because the neuron continue to tell the skeletal muscle cells to undergo contraction? Eventually, the calcium from the sarcoplasmic reticulum would be depleted. Once that happens, and the neurons continue to tell the muscle cells to contract, what happens? Will it contract? It can't contract because it ran out of calcium. Can it use the calcium from the ECM? No, there's no way, because there's no way to open up the calcium ion channel on the plasma membrane. You see that? So therefore, this is why skeletal muscle cells become fatigued, right? And again, we're not talking about the fatigue feeling from the buildup of lactic acid. We're just talking about literally if you use your skeletal muscle cells so much that you cannot contract anymore. Okay? So that's that is what happened. And again, hardwiring in terms of the calcium ion channel, the ryanodine receptors, which also is a calcium ion channel, is hardwired to be in the sarcoplasmic reticulum membrane and not in the membrane of the T-tubule. We're good? All right. So roughly, to give you an idea of how contraction works, is here's the actin microfilament right here, right? Microfilament would have binding sites. Here's the binding site for calcium as well as myosin head. But the myosin head binding site for the myosin head is covered or inhibited or covered by a protein called tropomyosin. So usually, if you have a myosin head right here, it can't bind to the microfilament to carry out contraction because that binding site is covered by tropomyosin. So remember, one way to regulate protein function is to inhibit the binding site, inhibit the binding sites, right? So that's it right there. So what calcium does is that calcium would come in and bind to the troponin complex, causing conformational change of the troponin complex, which in turns would nudge or push the tropomyosin out of the way, exposing the binding site for the myosin. Okay? And that's how contraction. So something to remember for contraction: calcium induces contraction, and ATP induces relaxation. Okay? So if ATP comes in now, then causes the myosin head to dissociate from the microfilaments, therefore you have relaxation of the muscle. Okay? So then if you have a flood of calcium in the cytosol, all of these troponin complexes along the microfilaments within the sarcomere will be bound by calcium. All of them will then cause the troponin complexes to nudge the tropomyosin out of the way. Then all of the myosin head that are near those binding sites would bind and then carry out contraction. That's why you have all or nothing. Again, hardwiring leads to all or nothing contraction. Don't worry too much about this. This is just saying that the myosin heads have to be regulated as well as also regulated by calcium. So the flood of calcium not only binds to the troponin complex, but also binds to the myosin light chain and then regulate the myosin head to be active, right? So that's skeletal muscle cells. So we talked about the fact that it's voluntary. We talked about the fact that it's all or nothing, and why why there is high power and why there is fatigability, right? Because again, the main source of calcium comes from the ER or sarcoplasmic reticulum, and that can be depleted. Okay? Versus cardiac muscle. What about cardiac muscle? It's spontaneous. It's involuntary. Okay. However, there's still a signal that tells it. It's just that the central nervous system is not controlling that. It is still all or nothing because the setup for the contraction is still the same, where there are sarcomeres and the same idea, the same setup as a skeletal muscle. It is at high power. You do want your cardiac muscle, your heart, to pump really hard, but you don't want it to fatigue. So no fatigability. So immediately, even if I don't say anything else, immediately in your mind you would think that you would know that the main source of calcium does not come from the sarcoplasmic reticulum, right? That should be the very first thing that comes into your head when we say that the cardiac muscle cells do not get fatigued. Okay? So how is cardiac muscle hardwired? Well, in terms of spontaneous or involuntary contraction, there's a thing called a pacemaker. A pacemaker is a group of nerve cells, right, that also receive information, so it still receive information, but the pacemaker would fire and would activate the cardiac muscles without your thinking about it. So it does that continuously, okay? So that's why it's involuntary, again, because of the pacemaker. Why it's also all or nothing because, again, calcium will flood into the sarcomere and bind into all of the troponin complexes, activating all of the myosin heads that are available, right? So that's all or nothing. Also high power. Why is it, so then fatigability. So where does the calcium ion come from, and what does that have to do with hardwiring? How cardiac muscles are hardwired is this, is the T-tubules themselves, right, still also have the DHP receptor. But in this case, the DHP receptor is connected to a calcium ion channel. Whereas in the skeletal muscle cells, remember the DHP receptors are not connected to a calcium ion channel. In this case, they are. Are there calcium ion channels in the membrane of the sarcoplasmic reticulum in cardiac muscle? Yes, but it doesn't matter, right? The point is that there's hardwiring, that the DHP receptors themselves are linked to the calcium ion channel. So therefore, if we move back, this is not exactly what a cardiac muscle looks like, but it's similar, where a cardiac muscle might be activated by the pacemaker, action potential occurs, moving down the T-tubule. But instead of moving on to the sarcoplasmic reticulum, what happened with cardiac muscle is that because the DHP receptors that are found in the membrane of the T-tubule are connected to calcium ion channels, that the calcium ion channels in the T-tubules will be open. And if you notice from this the histology here, that the T-tubule is opened up to the extracellular matrix. So where is the major source? What is the major source of calcium for cardiac muscle? The extracellular matrix. How much calcium do you have there? A lot. Calcium is stored in bones. Unless you have some kind of a bone disease, or if you're an invertebrate, you will have a lot of calcium in the extracellular matrix. Do you have unlimited amount of calcium? No. But there's so much that you couldn't possibly run out of calcium. And that is why cardiac muscles do not experience fatigability. Again, is because the main source of calcium ions come from the ECM and not the sarcoplasmic reticulum. Why are skeletal muscle cells, why do they experience fatigability? Because the main source of calcium ions come from the sarcoplasmic reticulum and not the ECM. So again, hardwiring. How are these muscle cells hardwired, right, then lead to whether or not they become fatigued? Yes, ma'am?
If the DHP is not connected to an ion channel in the skeletal muscle, what is its function?
It actually then it leads it allows the action potential to move into the sarcoplasmic reticulum. Right? Whereas for cardiac muscle, the buck stops there and doesn't move all the way into the sarcoplasmic reticulum. So again, since there are calcium ion channels in the membrane of the sarcoplasmic reticulum, you would think that they would be open, right? How are they open? By signal transduction pathways, and not by the action potential. Again, hardwiring. Does that make sense, right?
Yeah.
So, and then everything else is about the same. Okay. Did I double dip? Oh, I moved back, so okay, yeah. Okay. Smooth muscles. Smooth muscles are very different. Smooth muscles can form smooth muscle tissues, or they can be individual cells, right? So let's talk about these three things first. Smooth muscles are also, contraction is also spontaneous, so involuntary. So you can't directly control the contraction of smooth muscle cells. It's graded instead of all or nothing. So that means immediately you should think that when you hear graded contraction, you have to identify that by thinking about the fact that the level of calcium ions that leak into the cytosol is variable. Not only that, but not all of the myosin binding sites will be exposed at the same time, and not all of the myosin heads will be activated, right? So there's variation in all of that. So that's why it's graded contraction. Yes, ma'am?
Sorry, you said after the levels of the calcium ions are variable, what was after that?
So levels of calcium ions in the cytosol is variable. The number of myosin binding sites in microfilaments that are exposed are variable as well. And the number of myosin heads that are activated are also variable. Right. It's in terms of performance, it's anywhere between low and medium performance. So they will never undergo very high power, right? Or at least most of the time they won't. So it's low to medium, again, because of graded contraction. But there's still no fatigability. That means that again, immediately you should think that the main source of calcium ion does not come from the sarcoplasmic reticulum. In fact, for smooth muscle cell, it's about even, maybe half and half. Half of the calcium comes from the extracellular matrix. Half of it can come from the sarcoplasmic reticulum. There's a choice. If, let's say, for some reason, the sarcoplasmic reticulum runs out of calcium ions, smooth muscle cells can use calcium in the extracellular matrix exclusively as well. Right? So there's no fatigability. How does it work? How does a smooth muscle cell work? Well, here's how it works. Let's see. Let me describe to you, for instance, in your digestive system. Right? You have smooth muscle tissues that wrap around your digestive system. Okay? When you eat food and you need to push food through the digestive system, there's a process called the peristalsis or peristaltic movement. That actually the smooth muscle tissues do not squeeze all at once, right? They squeeze from here, and then lead to the squeezing here, and then so on. So they alternate. They take turns squeezing the food through the digestive system. That is called a peristaltic movement or peristalsis. So far, so good. All of that is controlled really by the original signal. So you don't have multiple signals telling different groups of smooth muscle cells contract, right? So how does that work? First of all, how are smooth muscles activated to contract? By hormones, growth factors, neurotransmitters, a number of different signals. So to regulate smooth muscle contraction, the smooth muscle cells have receptors that then have to be activated to activate signal transduction pathways. so no action potential is involved, right? So an example would be the activation of a receptor that activates PLC. PLC then would induce the expression or production of IP3, and IP3 can activate the opening of calcium ion channels in either the sarcoplasmic reticulum or the plasma membrane. There you go. That's how smooth muscle cells work. Okay, but again, the release of the ligand is not controlled by the central nervous system. Okay, so that's that. Let's skip all the way down to here. Is the fact that the amount of calcium ions is variable. So that depends on how many calcium ion channels are open, right? How many calcium ion channels are open depends on how many IP3 are being produced. So that's why, again, it's graded. Okay? Graded also means how many myosin binding sites in microfilaments are exposed. How is that controlled? As opposed to the striated muscles, as opposed to skeletal muscles and cardiac muscle, smooth muscle cells don't have tropomyosin. They have caldesmin, which perform the same function as tropomyosin. Caldesmin would cover the myosin binding sites. And so calcium, in addition to calcium then would be used for the solubilization or destruction of caldesmin. Okay, so the more calcium you have in the cytosol, the more caldesmin will be destroyed. The more caldesmin that is destroyed, the more myosin binding sites are exposed. So again, that's another level of variation. So the variability in the amount of calcium ions in the cytosol then leads to the variability in the destruction or degradation of the caldesmin, which leads to the variability in the number of myosin binding sites that are exposed. So that's graded contraction. In addition to that, the variation in the amount of calcium also induce the variation in the activation of myosin heads. Right? So not only do you have to expose the myosin binding sites, but the myosin heads have to be told to bind to the myosin binding sites. And not all myosin heads will be told to bind to the myosin binding sites. So that is also variable. How many myosin heads are told to bind to myosin binding sites is directly affected by two things. How much calcium ions are in the cytosol and how many myosin binding sites are exposed. They're all related. Let me run through all of that one more time for you. Okay? So the reason why smooth muscle cells have graded contraction instead of all or nothing is because of the fact that the number of calcium ion channels that are either in the ER or sarcoplasmic reticulum membrane or the plasma membrane, right, are variable. Okay, so the number of calcium ion channels that are open is variable, depending on what? Depending on how strong the signal is, right? And then depending on how many calcium ion channels are open, then that limits or that regulates the amount of calcium ions that are actually in the cytosol. The amount of calcium ions that are in the cytosol then will directly control how many caldesmin molecules that are being destroyed. How many caldesmin molecules are being destroyed directly control how many myosin binding sites that are exposed. The amount of calcium ions in the cytosol also control how many myosin heads, right, that will be activated to bind to the exposed myosin binding site. All of that is regulated, right? And there's variation, so it's not all or nothing, it's graded. And that's why, again, the hardwiring leads to graded contraction. The reason why smooth muscle cells do not experience fatigability is because the calcium ions that are required can come from either the sarcoplasmic reticulum or the ECM. And if the calcium comes from the ECM again, your body should never ever run out of calcium unless something is, you have some osteoporosis or something like that, right? Yes, ma'am.
Does different smooth muscle tissues, like the body, take it the calcium differently? Like does one area take more from the ECM and more take from?
No, that's a good question, but the answer is no. It actually depends on the signal. It depends more on, for smooth muscle cell, the variation depends more on the signals than if there's, let's see. So there is not a direct correlation between whether calcium is being taken from the ECM versus the sarcoplasmic reticulum. There is a direct correlation between the signal and how much calcium ions are being released. There you go. So far so good? Okay, now what about peristaltic movement? That has to do with the fact that within a tissue, smooth muscle cells have a lot of gap junctions. So imagine you have a lot of smooth muscle cells, right? A whole tissue. The smooth muscle cells that are on the outside are really the only ones that receive the information, the ligand, right? And then they would have an increase in calcium ion in the cytosol. But because there are a lot of gap junctions, guess what? The calcium ions not only are going to be used for this and this, but will flow down the concentration gradient through the gap junctions into cells that are in the middle, right? Not only that, but the same calcium ions will flow down this way. So that's why you have peristaltic movement, where these contract first, and then when calcium move into muscle cells further down the digestive system, then that's why they contract like this. Does that make sense? It's all due to, again, hardwiring gap junctions. It's all about hardwiring. Can you get, let's say, can you get smooth muscle cells to have fatigability? Yeah, if you hardwire it so that the calcium ion channels in the plasma membrane cannot be used, and only the calcium ion channel in the sarcoplasmic reticulum is used. Right? So and I don't know of, but there might be a disease when that happens, is that you might have a disease where something is wrong with your smooth muscle cells because of that, because the calcium ion channels in the plasma membrane are non-functional, for instance. Okay? So we do all of that. This is just a reminder where calcium. This I just dawned on me that this slide is in the wrong place. It should be further up. Okay. So this just explains the fact that each myosin head is being activated as individually. Okay. So that's smooth muscle cell. Again, going through all the smooth muscle cells, that is a way for me to integrate different information. And it is an example of convergent crosstalk, is the example of hardwiring, how the same function, right, can be activated by different ligand, different signals, and then different hardwiring allows for these different tissues to perform the same function, contraction, but in variable ways. Okay? So, last thing about for this set of exams is cell movement. Okay, we'll talk a little bit about. So you need to know this. This is really important, right? What type of cells move? Just about all types of cells move. How do they move? They can either swim, for instance, in the form of prokaryotic cells, in the form of unicellular organisms that are eukaryotic. And then your own cells can also swim, right? So they're swimming, and then there's crawling as well. Okay, and we'll look at very rough the molecular level of swimming versus crawling and what type of proteins are involved for different types of cells. Okay, so give you a survey of how cell movement occur. And by the way, cell movement, if you don't want to say cell movement, if you ever hear of the term motility, motility means cell movement. Mobility means movement of anything. So cell mobility is cell movement, but if you want to say motility, then that means cell mobility. Does that make sense? So motility, you don't have to say cell motility because that would be redundant. Even plants move. Plants are motile, right? And we'll talk about that. So strategy is swimming. Cells that have to swim needs a tail of some kind. So either cilia or flagella. Okay? Cilia or flagella. And they move through different means. So I'm going to expand on all of this. So prokaryotic cells, for instance, have flagella. Prokaryotic cells don't have cilia. They have flagella. But the flagellum is made up in the inside of proteins called flagellin. Right? And then how they move is they rotate. They rotate like this. Right? So if you imagine they rotate like this. So they rotate. If it's counterclockwise, if the flagella rotate counterclockwise, then they move forward. If they rotate clockwise, they just tumble. They remain in place. That's how most prokaryotic cells move, by swimming. That's how they swim. Prokaryotic cells don't really crawl. Okay, they swim, but they don't crawl. So that's it. That's very simple. That's all that you need to know about prokaryotic cells, is that they move by rotating the flagellum. They may have one or more flagella, and counterclockwise usually means that they move forward, and clockwise means that they tumble. How do they know to move, rotate clockwise or counterclockwise? By signals, by certain sources, right? We'll talk about chemotaxis in a minute. Okay? And why do they tumble? Because of, again, of the genetic molecular level has to do with the cellular level, because the flagellum for prokaryotic cells are supported by flagellin. Versus, don't worry about these, versus cilia and flagella for eukaryotic cells are supported by microtubules. So therefore, flagella and cilia for eukaryotic cells do not rotate. They whip around like a dog's tail. Right? Again, the variation in the motion has to do with the molecular structure. Right? Again, the reason why prokaryotic cells rotate is because it's connected with flagellin, versus the whipping motion is connected with microtubules. There's more to the story than that, but basically at this level, that's all that you need to know. Okay? So hypothetically, if prokaryotic cells have flagella that are supported by microtubules, how do they think they would move? By whipping the flagella, not by rotating. Right? Hypothetically, if you have a eukaryotic cell that contains flagella that are supported by flagellin, how do you think that these flagella move? By rotation. Right? So structure function. There it is again, right? Structure function. Okay? So that's very important. For cilia and flagella, again, like I said, they're supported by microtubules, and then very important. Okay, here's how flagella or cilia whip around is due to this. This is very important. Immobilized dynein. Where have we talked about dynein before? Dynein is normally used for intracellular movement, right? So dynein grab a hold of whatever it is and then walk along the microtubule, right? And in that case, for intracellular movement, dynein is free. But in flagella and cilia, the dynein is actually fixed. So the dynein heads are right here, and the tail is actually fixed, right? It's fixed onto another microtubule. So it's not free to carry material. It's actually fixed. So when it moves around like this, what does that? How does that work? If you have two microtubules like this, right, and the dynein is fixed, if it walks toward the minus n, guess what? What it does is that it bends the flagellum or the cilium to one side or the other, and that's why they whip around, right? Again, only because the dynein is fixed. Hard wiring, again, right? The dynein is fixed. The dynein is hardwired to perform this function to allow the flagellum and the cilium to bend, and the only way that the flagellum and cilium can bend is because the dynein is fixed. Okay. Again, just an example, there are unicellular organisms that have cilia, flagella. Cilia and flagella, they have multiple functions. Flagella allows cells to swim. Okay, they whip around and allows the cell to swim. Cilia do the same thing. Cells can swim around by whipping around cilia. Cilia can also have another function, can move the extracellular fluid around. So for instance, some of these unicellular organisms, let's say this is the mouth part right here, right? The cilia can move around to pull food material toward its mouth. So again, cilia are, but then these other cilia can be used for movement, for motility, for swimming. Okay, so cilia are actually more versatile than flagella. Flagella is purely for swimming, but cilia can be for swimming, but also can be for instance, gathering food. I think the next slide. In your trachea, cilia can move the fluid to. Remember about trachea, that the unwanted material is trapped in the mucus, but then the cilia can actually prevent this material from moving down into your lungs, right? So by whipping around, it does that as well. Okay. So again, having cilia doesn't always mean that the cell is motile. Having flagella always means that that's for motility. Okay, and cilia and flagella are supported by microtubules. They have immobilized or fixed dynein, and that's why they can whip around. Structure function. Other structures that are similar to flagella, for instance, the sperm tail, right? That has it's slightly more complex, but it still has microtubules. How about and that is also used for swimming. But how about exons, neuronal exons? They're not used for swimming, so we don't call them flagella, even though they're supported by microtubules. Okay. So again, I know that I'm showing you a lot of slides. I'm going through this fairly quickly, but the take-home idea is that flagella and cilia can be used for swimming. Okay, and if the flagellum is supported by flagellin, then there's this rotation movement. If the flagellum is supported by microtubule with immobilized dynein, then it's this whipping action. That's it. That's all that all these slides are. I'm trying to tell you, okay? And giving a lot of examples. 9 plus 2 array. If you learned this from somewhere, fantastic. If you haven't, don't worry about it. This is just to tell you how complex it is. Right? So here's the diamine that is fixed. It's going to walk. This is a cross section, so it's going to walk along this microfilament, microtubule here, and then again causing the the flagellum or the cilium to bend either one way or the other. That's it. Okay. So don't try to memorize these structures. Again, same thing here. So it's fixed at the tail, and then the head would walk, and as it walks toward the minus end, this structure bends, right? And that's why, and it bends back and forth. So it bends back and forth. And that's how materials either get moved around or cell motility or motility occurs. Okay? Yes, I'm saying the same thing 5-6 times. This is what we're going, the main thing we're going to focus on for the next half an hour, 35 minutes. Okay, is cell crawling. Cell crawls right off the bat. And when you hear about crawling, it involves microfilaments and myosin. So it involves contraction. So there are multiple things that we have to talk about. We have to talk about how it's triggered, how crawling is triggered. We have to talk about the actual contraction itself. We have to talk about interaction or anchoring onto the extracellular matrix. And then we have to talk about the cell shape as well. Okay? So all of that is involved. So again, this is an example where there's integration of information. Okay? Pretty picture remind you about, what does it remind you about? Okay.
Embryo development.
The filaments.
Pretty picture. Chemotaxis. Here's the idea. Okay? There are these two kinases. One is called RAC, and one is called RHO. RAC and RHO, get it? Okay? They are both required for crawling. And these two slides right here just tell you that RAC and RHO are activated, and pay attention to the activity here. So don't worry about this part, worry about this part. So for RAC, what RAC does is that it acts at the leading edge. So if a cell is moving toward in a certain direction, that's the leading edge, right? And RHO acts at the trailing edge, which is back here. RAC also induces actin microfilament polymerization and disassembly at the leading edge, only at the leading edge. RHO regulate the same activity at the lagging edge, right? So now one acts at the leading edge, one acts at the trailing or lagging edge. And then both of them also, so both of them do this, have the same activity. Again, only that RAC is doing everything at the leading edge and RHO does everything at the trailing edge, okay? But both of them have to be activated. That's all that's saying, okay? So far so good? All right. So let me describe how this works. How does a cell know to move in a certain direction? By something called chemoattractants. All right? So there are chemoattractants are signals, ligands, signaling molecules. That the chemoattractants can be food source. Can be a food source. Chemoattractant can be, let's say, a certain toxin or certain molecules that's secreted by the bacterium, and this cell could be an immune cell that is drawn to the chemoattractant so that it can attack the bacterium. So there's some kind of signal there that attracts the cell toward it. Okay? The chemoattractant then can make its way to the receptor, surface receptor, which then can activate either RAC here or RHO here. Okay? So far so good? And let's say that this cell is, before all of this happens, it's just sitting there anchoring onto the extracellular matrix. When it's anchoring onto the extracellular matrix, it's anchoring through integrin. And chances are, because it's not moving, the integrin on the cytosolic side is anchored onto the intermediate filament. Because it's just sitting there, right? So then the idea is that this signal will activate RAC, and one of the things that would occur is that the integrin then would switch from anchoring onto intermediate filament to anchoring on to the microfilament. And that allows the cell to know that, okay, we have to move, right? Okay? So far so good? All right. So movement is this. So again, another thing is, and I'll go into more details. Movement is when this plasma membrane at the leading edge would extend out. And you've heard about the term lamellipodia, filopodia, false feet, right? That's what they form, okay? So this, at the leading edge, the plasma membrane extends. How does it extend forward? By polymerization of microfilament. Polymerization of microfilament extends. Polymerization of microfilament is regulated by what? By RAC. RAC is regulated by the activation of the receptor. The receptor is activated by the chemoattractant. Right, so chemoattractant or signaling molecule activate receptor, which activate RAC, which then induce polymerization at the leading edge. Okay? And then in order for the cell to move, what happened is that the leading edge would extend out. The body right here, right, either at the leading edge or at the trailing edge, would undergo contraction. So the myosin filament and microfilament would pull against each other, and contraction occurs. And so contraction similar to smooth muscle contraction, not the same thing as the skeletal muscle and cardiac muscle contraction, but more similar to smooth muscle contraction. Okay, and it's through the stress fibers. Okay, and then at the lagging edge, then so now you have contraction and the cell is elongated, but you don't want the cell to elongate. You want the whole thing to move, right? When you walk, you don't want your tail end to drag along the floor, right? You want the whole thing, your whole body to move. So then the trailing edge would have to pull forward. Okay? So you have to have disassembly here. And in this case, disassembly is actually at the plus end of the microfilament. You see why we talk about the fact that disassembly either by depolymerization or severing may occur at either the plus end or the minus end, right? Because sometimes you do want disassembly, but at the plus end instead of the minus end. So far, so good? Okay, so that's how cells walk. Let's look into it in further detail. See, the idea is this, okay? Normally, when the cell is in place, you have rack and roll, and rack and roll is distributed throughout the cell. Because your question might have been, and nobody asked, but question might have been, well, how does the cell know to put all the racks up in this side and all the row? What happened is the chemoattractant is right here, right? So the cell doesn't know. So that's why the cell spread everything throughout. Rack and roll are found throughout the cell, okay? And whenever you have some kind of a signal, then only these racks are activated and these rack kinases are not activated. So when these rack kinases are activated, you know that this is the leading edge. The reverse is true. Only the row kinases are activated here and not here. So therefore, when the row kinases are activated here, then you know that this is a trailing edge. Right. Again, all of this is hardwired. We actually have pictures of this, right? So this is lamellipodium right here, and you notice that also remember where can microfilament, where are fine microfilaments located in the cell? Mainly in the cell cortex, right? And then if a cell is sitting in place, then there's stress fiber underneath. So the stress fiber is what can undergo contraction. But the extension into lamellipodium and filipodium is actually through the the polymerization of assembly and disassembly of the microfilaments in the cell cortex. Right? It's all in the cell cortex right here, and that's what it's showing here. So more details about things that you already learned. So what I went through so far is the mechanism of roughly how migration occurs. Now let's look at the actual regulation of migration. Right? So here's a microfilament within a, let's say this is the leading edge, and here's the cell cortex, and this is microfilament in the cell cortex. Okay? This is a plus end. Okay? Then what happened is that ATP-bound G-actin is going to be added toward the plus end so that polymerization can occur, so that the lamellipodium can be formed, right? So the plasma membrane can extend into the lamellipodium, and obviously profilin will then makes the whole process more efficient, right? At the minus end of this microfilament, the trailing edge is all the way out here, right? This is not the trailing edge here. This is just the nucleus is somewhere right here. So this is still the leading edge, but toward the minus end of the microfilament within the leading edge, right? Depolymerization is going to occur. Because why? Because you would have ADP-bound G-actin in the microfilament, and then cofilin will remove the ADP-bound G-actin from the minus end, right? And so the idea is that as the cell moves forward, you don't want to vary the size of the microfilament. The cell size needs to remain the same, but then the length of the microfilaments will also have to remain the same. So then the cell will remove the G-actin from the minus end and put it toward the plus end. In the en route, the ADP that is bound to the G-actin is going to be switched out for an ATP. So you need a lot of ATP toward the leading edge. This right here, this process right here where ATP-bound G-actin is added at the plus end and ADP-bound G-actin is removed, we call this treadmilling. Like if you walk on a treadmill, right, or a tank, the same G-actin is being used, but they're just being recycled, right? So the same G-actin, so this is still the leading edge, right? This is the cell cortex right here, and so these G-actin is going to be moved up here, right? And en route, the ADP is going to be exchanged for the ATP. Again, we call that treadmilling. Why is that important again? That is to ensure that the length of the microfilaments remain the same, unchanged. Again, this is the leading edge, right? This is not the lagging edge. This is the leading edge. Okay? So that's the very first thing, a cell that needs to move. I will explain this in the following pictures. Okay? So rack and roll activate these signal transduction pathways leads to these effective function. Just to give you an idea, don't try to memorize these two slides. Just to give you an idea that signal transduction pathways are involved, and then effective functions are involved. Okay? Don't try to memorize them. So add complexity to what we already talked about in terms of treadmilling. So if we were merely talking about the microfilament, that's what happens. But what is a function of microfilament? Microfilament is is used as an anchor for integrin. Right? So you would have a bunch of integrins at the bottom of the cell, right, anchoring onto the extracellular matrix, and that's how they walk, is by still anchoring onto the extracellular matrix, right? Anchoring on through the extracellular matrix through integrin. So here's the deal: here's the integrin right here, and as a new ATP-bound G-actin is added, integrin then will also continue to anchor onto the growing microfilament to ensure that the cell continues to be anchored onto the extracellular matrix. What happened at the other end when you have the ADP-bound G-actin being removed? If ADP-bound G-actin is removed, what happened to that integrin? No more anchorage at the cytosolic site. When you don't have anchorage here, what does that tell the non-cytosolic site to do?
Stop.
To not anchor onto the extracellular matrix. So again, that idea of linked binding is really important. If there's binding here, then if there is binding here, then there's binding here. If the binding here is removed, then there's no binding here. So that allows the cell membrane to be dissociated from the extracellular matrix. So that's how cells move, right? So again, as a cell moves, then the plasma membrane cannot afford to interact with the extracellular matrix any further because it needs to move forward. Because if it's still anchored on, what happened to the cell? It gets longer and longer and longer. Does that make sense? Okay, tracts. So all of this, what you pay attention to is this term tracts. When a cell moves, it moves in one direction. It doesn't move crisscross like that. And what it tends to do is, let's say it wants to move in that direction, and the collagen fiber is this way, right? But the chemoattractant is over there. So what the cell does is that it doesn't move along with the collagen fibers. Guess what it does? It makes its own road. Right? Remember, the idea of a cell will synthesize and secretes its own extracellular matrix. That's what it does, is that if it needs to move there, it's just going to make its own collagen fiber. So we call that a tract. Right? A tract. Okay. Lots of different ideas all integrated into the same process here. More. As if that's not complex enough, the idea of what about other cellular components? Right? What happened to other cellular components? As a cell moves forward, it has to move mitochondria and lysosomes and vesicles and everything forward. So myosin, the molecular motors, have to be involved, right? Again, both at the leading edge and at the trailing edge. Okay, so leading edge, again, leading edge move forward, contraction in the middle, and then trailing edge would also be pulled forward, and it's pulled forward by again when the ADP-bound G-actin is removed by cofilin, that the integrin will no longer interact with the extracellular matrix, therefore the plasma membrane in this part will lift up, right? At the same time, cellular components will have to be moved forward through myosin motors, okay? What about the ATP? At the leading edge, there's a lot of ATP required, right? Why are they required? Because of that exchange from an ADP bound to an ATP bound. Lots of ATP. Where does ATP come from? either glycolysis or the Krebs cycle and cellular respiration. Well, is it easier to, if you need 1,000 ATPs, would you want 1,000 ATPs to be moved forward, or do you want to just move mitochondria toward the front, and then the mitochondria can produce as many ATPs as required? And that's actually what happened. Is that mitochondria are actually moved, are directed to move forward, but not along the microfilaments. It will be along microtubules. So then, at the same time as, again, the lamellipodium is being formed through polymerization of microfilament at the leading edge, contraction occurs in the middle, right? And then at the trailing edge, then depolymerization occurs, and the membrane gets lifted up. All in the while, the microtubules would also be reorganized so that a lot of them will point toward the leading edge. To do what? To deliver mitochondria toward the front so that a lot of ATP can be produced. Yes, ma'am?
Does it always start with the forward movement of RAC?
It has to start with that, yes. Right. That's what gets everything jump
And
started.
then row.
Right, right, yes. So far, so good. I know that that's a lot of information. The reason, and so I always tend to have reason, even though it's my own reason, but it's a reason. When I went through that, all of that, I didn't slow down is because you have heard all this story before, right? The only thing I'm doing is integrating everything that you've learned into cell movement. So I didn't tell you anything anything new. The only thing I did was integrating information, right? However, having said that, I will spend a couple of minutes if you want to ask questions about cell movement by crawling. Are there any questions? Or is that just so fast that you don't even have a question? I saw a lot of nodding heads and smiling. Apparently, some people got it, and then it's, well, some people got it. Yes, ma'am?
Is RAC and row activated at the same time, or is RAC activated first? And then
Roughly at the same time, roughly. And so a better question to ask is, are they always, do both of them have to be activated? And yes. If one is activated, the other one is also activated. It wouldn't do the cell any good to extend forward and the cell just stretches out, right? Yeah. Okay? Using this as a jump start, as a bridge into what we already talked about in terms of muscle contraction. Okay? So what on earth does muscle contraction and cell migration have in common? Well, obviously contraction is in common, right? Because cell migration requires contraction. Muscle contraction is contraction. But another thing that is important that we haven't talked about is the requirement. In order for muscle contraction to work, in order for cell migration to work, they both require the same thing. Okay, again, we're talking about both cell migration by crawling and muscle contraction have to do with contraction, right? But for both activities, in order for contraction to occur, you have to have some kind of friction, some kind of anchorage. If you have your muscle cells contracting, but it's not hanging on to anything, right? So if my muscles are contracting, but it's not hanging on, if there are no tendons, no bone, thank you, right? Then I can't move, right? So there has to be friction, right? The tendon acts as, basically as anchorage for friction. Same thing with cell crawling. If the cell extend and then contract, but if it's not anchored onto the extracellular matrix, guess what? It just does this in place, right? It just does this in place. So the extracellular matrix is very important so that when contraction occurs, that there's some pulling and pushing activity, not pushing, but there's some pulling activities occurring. Okay? And it is a pulling activity. It never is a pushing. Contraction, so just like cells never extend, right? They contract. They contract and pull. So pushing and extension is never involved with contraction, right? So it's always contracting and pulling, okay, for both muscles and cell movements. That's what these slides refer to. Okay. And last bit. Are we good so far? Last bit about plants. I think only 2 slides. 2 slides. Plants actually have motility, but plant motility is regulated differently. Okay, how it does it is different. Mainly because plants stay in place. So motility or movement has to do with the entire plant changing its shape, right? The entire plant, not just individual cell, but entire plant. So there are 2 types of movement, plant movement. One is called nastic movement, and one is called tropic movement. Don't try to memorize all of these. You do need to know the difference between nastic movement and tropic movement. Okay? Nastic movement is when it has to do with osmotic pressure. Okay? So, for instance, in this particular case, this Venus flytrap, this plant, is a carnivorous plant. Would normally the leaves would be like this, and then it will close, right? It will close. Where did the signal come from? The signal comes from the interaction between the plant cell and the insect. That's where the signal is, right? It closes. How does it close? By changing osmotic pressure, by the change in the amount of water in the central vacuole. By putting more, let's say it's like this, okay? So for some of the cells, if you remove some of the water, amount of water in central vacuole, it will wilt, and it does this, right? And then if you want to open, then you have a higher flow of water into the central vacuole, and everything will extend out, right? So it's a change in the osmotic pressure within the plant cell. So that's nastic movement, again, literally changing the shape of the plant. That can be guided by touch, like this. That's what it's called. By light, by both light and darkness, by chemicals, in response to water, gravity, temperature, pH sometimes. Okay? So again, nastic movement is the actual change in the shape of the plant, versus tropic movement is cell proliferation. We also consider this to be plant movement, even though, again, the cell doesn't move. But if you have gravity, in this case, this is a case where you have both gravitropism and water is hydrotropism. So there's water here. There isn't water here. So the plant cell, what it does is that it grows. It detects moisture here, right? And as well as in conjunction with gravity. And that is enough signal to tell the cells in the root here to undergo cell proliferation and grow toward the source of water. Okay? So tropic movement is plant movement through cell proliferation, versus nastic movement is plant movement through changes in osmotic pressure. Okay? So again, it has nothing to do with talk about my microtubule or crawling or swimming or anything like that. Alright? Actually, 5 whole minutes for question and answer.
Is it still working?
You just
Yeah.
tired, you want to go home and just. You can come on up here.
Oh, that's good. Ma?
I don't want to help.
Very